Welcome to the course G08, and here we're at video 2 on metagenome sequencing. So, there's different sequencing approaches for metagenomics, and tells much it depends on what we're trying to do. So we can take our samples from different areas, whether it's from vertebrates of interest, marine creatures, soil, whatever it is, water. There's many different types of sampling, and we can do minimal prep to sequence that, extract the metagenomes and to an extent the metatranscriptomes from that. Or we can do more lab-based tests to do some isolation and identification of specimens that are of interest, to then do sequencing to extract genomes that are of interest from either approach. So we're dealing with known genomes or metagenomes. Either way, what we can do with this data is kind of inform our molecular biology hypotheses that are of interest to us in this context. We can, in certain contexts, look at traditional pangenomics using genes. However, for viruses, that may be a quite limited approach. And we can also look at the relative level of the different viral specimens as well. There's quite a lot of different sequencing approaches that are worth considering. That we can differentiate metagenomics here from things like amplicon sequencing and other approaches based on the protocol that's being used. So all of them, we have DNA extraction here for amplicon sequencing. We used targeted enrichment approaches. Often that now is using some probe-based method, but we're targeting certain regions to sequence. Whereas if we're using here metagenome approaches, we're trying to remove any biasing steps in the sequencing process wherever possible, if it's sensible. It depends on the exact idea being explored. This is different from metatranscriptomics, where the RNA is extracted and sequenced, and same with the metaproteomics, where we're looking at the protein data at the start. So doing protein extraction, digestion, mass spec to get more information on that. And same with metabolomics, we're looking at metabolites with mass spec or NMR to get information from that. So these methods are complementary but different. An example of what we can do with metagenomic data is take a metagenomic sample that's of interest, extract the DNA, shear it in and transfect it into plasmids that can then be grown in cell line, for example, and then we can do functional metagenomic selection to sequence certain ones that are of interest, to identify ones that have a phenotypic effect on the cell line or sample that's of interest. So there's a lot of things that we can do if we're going to discover more about different viruses and the genes that they have and their effects on viral phenotypes. Another more complex workflow is shown here. So here we have maybe a sample source, which is a stomach in this case. We can go to sample collection and extract the metagenome from that to identify what viral organisms are there. We can do QC, let's do assembly or co-assembly, taxonomically identify what is there, and map them to the annotated contexts that are of interest to see what's there. We can also map the reads to known genomes as well to identify if we have the correct ones. We can also do de novo assembly to discover more about novel genes that may be present, and then do standard pathway analyses to identify changes at the pathway level that might imply certain K groups or GO categories in the novel phenotypes that we see. Again, that's more coming from the bacterial point of view. An example of this is something that you all well know. So we have on the 16th of December 2019, early in the morning, there's metagenomic sequencing of some sort of novel virus that had 81% similarity based on this very limited sequencing of it, just containing a few reads of something somewhat related were distantly related to SARS-1 and also 85% of similarity, give or take, to some bat SARS virus that they were trying to explore that morning. So they're able to compare to existing coronaviruses and create a phylogeny of that to identify to what extent was it related. And this was just based on the initial sequencing that was kind of a test one for the higher coverage sequencing that they did later. So you can see they were able to gradually over time get more information on this, including a tree identifying that there was some unknown coronavirus that had this phylogenetic position that was quite conserved in subsequent analyses. And you can see here that they did de novo assembly to identify the genome length of being just under 30 kb, which is a typical beta coronavirus length, and to extract good context from that to explore better its biological properties. So this is that's kind of standard things you want to do with metagenomics. You have partial information, perhaps, and you might want to assemble it to identify what's its evolutionary origins. And what was useful here is that on the 30th of December, a competitor, in terms of that's what the term used in the translation, detected the same virus in another patient's sample. And they found that this sample was also distantly related to a SARS coronavirus. And later that day, that was announced more widely, and I think certainly in the Ireland and the UK, by December 31st, that was widely available on social media, not just in China but globally as well. So you can see how if we have rapid metagenomic approaches, that can be useful in terms of communicating what may be present and acting on that rapidly. So there's more at this link if that's of interest to you. So that concludes this video. Please see more on metagenomic classification with Kraken 2.